I see you're in front of Radcliffe camera.
That's right. That's right.
Yeah.
Sunny day.
But you know, sun comes from different direction to you than the Radcliffe camera.
Not in Oxford. We have these mirrors, you know.
Thanks for coming here today, Ricardo.
Thank you for having me. Thank you for the invitation.
It's a pleasure to have you here. Your paper created quite an enthusiasm in immunology field, but also in general membrane field. We are very excited to hear about your paper.
Great.
But before you start sharing your slides, could you please introduce yourself with a few sentences and then start sharing your slides?
Yes, absolutely. Well, first, thank you for having me. It's a great pleasure to be here. I'm Ricardo Fernandes. I just moved recently back to Oxford, where I did my DPhil. Now I'm starting my own research group here. And before that, I spent 5 years at Stanford working in the lab of Chris Garcia, where I did my postdoc. And both in my DPhil and in my postdoc, I've been working on trying to understand receptor signaling, mainly focus on receptors that are expressed in T cells. And more recently in ways that we can use to try to modulate their signaling.
Great. So start sharing your screen then, and stage is yours whenever you want.
Thank you. Okay, let me just get this fired up.
Looks great.
Great. Can you see the laser pointer as well?
I do see, yeah.
Okay, fantastic. So I'd like to start with this first slide because it really reflects in a very simple way what I'm interested in and what the focus of my research has been, which is to first try to understand the signaling mechanism by many different surface receptors, mostly receptors that are present on T cells. And then more recently in finding ways where we can use this information, so the signaling information, but also structural information, to guide us in the development of new tools that allows us to explore not only their function, but also to change their function in particular settings. Now, as many of us know, T cells use a lot of many different receptors to recognize pathogens, mutations, but also self. And I'm just showing here an example of a few proteins involved in these processes. Of course, the recognition of these different foreign or self peptides and agents plays a big role in infection, but also in cancer and in autoimmunity. And the talk that I'm going to be presenting here today is mostly focused on cancer, and in a class of receptors such as PD-1 and CTLA-4, which provide an inhibitory signaling in T cells. So here, essentially, what I'm trying to show with this cartoon is that even though receptors such as the T cell receptor, CD28, PD-1, and CTLA-4, they have very different structural architecture, they actually signal in a very similar way. So the T cell receptor and CD28, they have what it's called activating motifs. So these are two tyrosines present in the cytoplasmic domain that when they become phosphorylated, they initiate a signaling cascade that activates and promotes activation of T cells. On the other hand, receptors like PD-1 and CTLA-4, they actually inhibit signaling. They reduce signaling by T cells. So when T cells are exposed for longer periods of time and when they produce a sustained activation, they also start expressing these inhibitory receptors as an attempt to reduce signaling. So particularly in cancer, what we would like to do would be to essentially shut down or eliminate signaling by these inhibitory receptors. And why? That's because the aim would be to maintain robust T cell activation instead of having a decreased response over time. So how can we modulate signaling by receptors? There's been mainly two different ways of doing this. One is to engineer natural ligands, and this has been done, for example, for cytokines. And that's usually done with soluble ligands. So it can't really be done for the PD-1 or CTLA-4 because they bind receptors that are present on other cells. So to change the ligands, we would have to change the cells too. So that's not really possible here. Another approach is to use antibody blockade. And this approach has also been used for many different receptors, for growth factor receptors, also for cytokine receptors, and for immune receptors. The idea of checkpoint blockade is very simple, and I'm just showing here a very simplified cartoon. So if we have a receptor that binds to its natural ligand and initiates a signaling response, then what we can do is we can engineer antibodies that bind to the receptor or to the ligand and essentially sterically prevent them from binding each other as an attempt to prevent signaling from occurring. So that's what we mean by checkpoint blockade or antibody blockade. Now, of course, checkpoint blockade has been revolutionary in immunotherapy because quite a few years ago now, almost 20 years ago now, there's been antibodies that were developed to bind to CTLA-4 and block this receptor from binding to its ligands. And the same thing has been developed for PD-1. And this has led, of course, to the Nobel Prize to Jim Allison and Honjo a couple of years ago for their pioneering work on this. And also, not just because of the mechanism itself, but also because a lot of these antibodies were later used in the clinic, and they were proved essential in reducing tumor progression in many different types of tumors. And this has really changed how immunotherapy is done today. So as you can see, some of these first antibodies, checkpoint blockade antibodies like ipilimumab, were developed in 2000. They were then later approved after 10, 12 years. They were approved for melanoma. And now a lot of these antibodies, checkpoint blockade antibodies, have been approved for multiple different types of cancer. Now, unfortunately, despite some patients benefiting from these treatments, most patients still fail to respond to this type of treatment. So we wanted to see what we could do in terms of reducing signaling by these receptors. So for that, we wanted to essentially give one step back and first look at the mechanism, or try to see what is the mechanism that leads to receptor phosphorylation, what initiates signaling by these receptors. So I'm just showing here again, for all these receptors, like the T-cell receptor and co-receptors, signaling these receptors do not contain a tyrosine kinase associated with them, like it happens for cytokine receptors or for growth factor receptors. They instead rely on phosphorylation from kinases that are present on the membrane, which are part of a family of Src kinases. They're not directly associated with these receptors, and they are present at the membrane and they're active. So they can phosphorylate multiples of or many of these different receptors. Also, it has been shown for the T-cell receptor and for other receptors that signaling can actually occur in the absence of ligand binding. It's also called tonic signaling. So these receptors can signal even before, or they can become phosphorylated even before binding to their ligands. So we just thought of a very simple question. Is it the case that tonic PD-1 signaling or CTLA-4 signaling contributes to T-cell exhaustion? So can it happen that signaling by PD-1, even before it binds its ligands, PD-L1 or PD-L2, can it inhibit T-cell activation? So to test this question, we just started with a very simple experiment. So we took Jurkat cells. These are T cells that we use frequently. We just used these Jurkat T cells and we CRISPR'd out PD-1. And what I'm showing here is just a very simple activation of Jurkat T cells with this antibody called OKT3. This antibody binds to the TCR and activates T cells. And as you can see at the start of the experiment, this is an overnight experiment. At the start of the experiment, if cells are not activated, there's no PD-1. But then when cells become activated with OKT3, you can see in the wild type, the blue line in Jurkat cells, PD-1 expression goes up. Now in the knockout, we don't see the same type of PD-1 upregulation, which is what we wanted. And then we asked what happens in terms of activation now that we have this CRISPR PD-1 cell line. So what we can see is that if we look at CD69, which is a marker for activation in T cells, then you can see that wild type Jurkats, after activation with OKT3, overnight, they will upregulate CD69. But the Jurkat T cells where we CRISPR'd out PD-1, they have a higher level of activation. So it looks like not having PD-1 can lead to stronger, more robust activation. It's also important to mention that these Jurkat T cells, they don't have PD-L1 or PD-L2. So in theory, there should be no ligands here for these PD-1. Now, if we treat these cells with nivolumab, which is an antibody that binds to PD-1 and blocks it, so prevents it from interacting with PD-L1 or PD-L2, we see no difference. So again, here we're just showing wild-type Jurkat cells treated with Nivo. You see this upregulation of CD69. If we treat the Jurkats where we CRISPRed out PD-1, you see the same level of activation as before. So ideally, what we would like to do would be to essentially push this level of activation from the wild-type Jurkats to a situation where PD-1 is absent. But it looks like binding with nivolumab is not enough. Of course, this is not surprising because there's nothing really to block here because the ligands are not present. Next, we did the opposite experiment, which was to overexpress PD-1. So I've shown already before the red line, which is the activation level that we get when PD-1 is not present. In blue is the wild-type Jurkat cells, and then in green is when we upregulate or we overexpress PD-1. And as you can see, overexpression of PD-1 drastically reduces activation. So again, it looks like this activation level that we're observing by measuring CD69 is dependent on the levels of PD-1 that are present on the cells, even though there's no ligand. Now, I keep saying that there's no ligand, but of course, maybe there is a ligand in these cells that we're not aware of. And this, of course, could happen. So what we decided to do was to generate this construct where we truncated the extracellular region of PD-1 and swapped it for an HA tag. And we just use an HA tag for detection at the cell surface. So here what we have is we have wild-type PD-1 and we have truncated or ECD-less PD-1 that still has the native transmembrane and the native intracellular region. So it can still signal, but now it shouldn't be able to bind to anything. So when we test the expression of these, or the effect of the expression of these receptors, what we see, looking at CD69 and activating cells with OKT3, is that even when we express this truncated form of PD-1, we can still see a decrease in activation levels. So again, this is just to point out to the fact that it looks like expression of the intracellular region of PD-1 is enough to drive a reduction in T-cell activation. We also wanted to see whether we could detect phosphorylation of PD-1. So for this, again, we overexpressed PD-1 in these cells. And even in the absence of any stimuli, we just IP'd PD-1 from the cell surface and looked at phosphorylation. And again, you can see here, we detected some weak levels of phosphorylation, which again suggests that there is some tonic signaling of this receptor in T cells. And we can see that in terms of phosphorylation, and also we can see it further downstream when we look at markers of activation, such as CD69. So it looks like the initial picture that we drew before wasn't really complete, because not only we have signaling being initiated by the ligand, we also probably also have a ligand-independent signaling going on. And that's potentially because of the signaling mechanism that these receptors employ, which is they rely on these kinases that are present on the membrane that are active to phosphorylate. And so it's possible that these kinases, even if weakly, phosphorylate the motifs, the signaling motifs present on these receptors. So then the idea would be that if we were just to use the antibody blockade, this of course would probably be more than enough to prevent ligand-induced signaling, but maybe wouldn't be enough, or it looks like it's not enough to reduce the ligand-independent signaling. So we went back again to our drawing board and we start thinking, how can we address this tonic phosphorylation? So luckily, T cells, they express at the surface phosphatases, such as CD45. And I say it's luck because these phosphatases are extremely promiscuous, such as CD45. And what I mean by that is that they can dephosphorylate many different receptors. They're highly abundant. Almost 10% of all the proteins at the T cell surface are CD45, and they're also active. So we thought, can we essentially hijack the activity of these enzymes, of these phosphatases, and bring it to the vicinity of receptor to drive receptor dephosphorylation? And that's what I'm showing here. So we thought we could generate these molecules, these bispecific diabodies, that would bind to CD45 and to PD-1 in cis in the same cell, recruit CD45 to the vicinity of this receptor, and essentially locally force an increase in the phosphatase activity with the aim of dephosphorylating the receptor. And we can also engineer this in a way that when these bispecific diabodies bind to PD-1, they also block PD-1 from binding to its ligands. So the aim was to achieve a dual effect, not only block binding, but also promote dephosphorylation. So that was the aim. And we started by making these bispecific diabodies essentially by scanning the patent literature and trying to find sequences of scFvs that would bind to these molecules. So here I'm just showing the purification of this protein. It looks good by size exclusion chromatography. Here is the gel. By SPR, we confirmed that this protein was indeed binding to CD45, human CD45, and also to PD-1. Now, the scFv for PD-1 is the scFv from nivolumab, which is the antibody that has been used in the clinic. It binds with much higher affinity to PD-1 than with CD45. And this was a complete random chance that we found these scFvs, but this is exactly, I think, the type of binding that we want. We want the binding to be mostly driven by the receptor and after by the phosphatase and not the other way around. But of course, we didn't know whether these molecules could bind both molecules simultaneously. So to test for that, we used what I call the sandwich Biacore, in that we immobilize PD-1 on two separate channels. And here I'm just showing immobilization of PD-1 in these two channels. In one of the channels, we flow RIPR, which binds to PD-1. And then in both channels, we inject CD45, soluble CD45. And as you can see, in the channel where there's no RIPR, we detect no binding of CD45. In the channel where there's RIPR, we can detect CD45 binding. So it looks that, at least in solution, these molecules are able to bind both to PD-1 and CD45 at the same time. We then moved to a different type of experiments. And here, what we wanted to see is whether these molecules can actually induce dephosphorylation of PD-1. So for this, we took HEC3 cells, and HEC293 cells, they don't express any of these receptors. They don't have PD-1, LCK, or CD45, which essentially gave us the chance of reconstituting PD-1 signaling in these cells in a very simple form. So here, the data that I'm showing is for PD-1 phosphorylation after IP for PD-1. And in all of these lanes, PD-1 is present. LCK is only present in lanes 2, 3, 4, 5, and 6, so it's not present in lane 1. And this is why we don't detect PD-1 phosphorylation. When we express LCK, we can see strong PD-1 phosphorylation. If we co-express CD45 and LCK, you can immediately see a reduction in PD-1 phosphorylation. So this is the equilibrium that we have in terms of phosphorylation between having a kinase and the phosphatase in these cells. And then we incubate these cells that express the three proteins, PD-1, LCK, and CD45 with RIPR. And as you can see, incubation with this RIPR PD-1 molecule leads to a dephosphorylation or to a reduction in PD-1 phosphorylation. So it looks like these molecules can dephosphorylate or can induce PD-1 dephosphorylation. However, we're also dimerizing these two molecules, of course. And we were concerned whether some of this effect that we're seeing is actually due to changes in protein dynamics, changing in protein diffusion. So, and in a way, whether this could be almost independent of the phosphatase, which could be the case. So to test for that, what we did was to express, instead of wild type CD45, in lanes 5 and 6, we expressed the form of CD45 that is CD45 dead. It contains a single point mutation in the active site such that the phosphatase activity is not present anymore. And as you can see, when we incubate these cells with RIPR, now we don't really observe a decrease in PD-1 phosphorylation. So it looks like to observe this decrease in phosphorylation, we need an active phosphatase. Binding to the extracellular region is not enough. So this was all done in HEX. So then we moved to working with actual T cells. So the data that I'm showing here is again in Jurkat T cells that were activated with a fixed concentration of OKT3, which is going to bind to the TCR and activate these cells. We're looking at 3 different markers of activation, CD69, CD25, and IL-2. And we're treating these cells either with nivolumab, the antibody that binds to PD-1 and blocks PD-1 from binding to PD-L1, or with this RIPR, the bispecific CD45 PD-1 molecule. And as you can see, in the absence of any treatment, we have this level of activation here shown with a black dot. When we treat these cells with increasing concentrations of nivolumab, we don't really see an increase in activation. But if we treat these cells with the RIPR PD-1, you can see a robust enhancement of CD69, CD25, and IL-2. Now, again, just to say that nivolumab, obviously, we weren't really expecting nivolumab to work under these conditions because there's nothing to block. But it also shows that RIPR, even in the absence of ligands, it can still generate a potentiation in T cell response. Now, this was done in jerkets. So we wanted to move to actual PBMCs or T cells isolated from PBMCs. So what we did here was to take T cells from healthy donors and then stimulate them with a cocktail of peptides. These are close to 200 peptides for things like CMV, influenza, EBV, which a lot of the population has and can respond to. So we incubated T cells with this cocktail of peptides, and then on top of that, we treat them either with nivolumab or with RIPR PD-1. And then again, we looked at markers of activation such as CD69, CD25, and interferon. Now, in this mixture of cells, there's also cells that will present PD-L1 and PD-L2. So here, the ligand for PD-1 is present. And as you can see, when we have cells stimulated, if we treat them with this cocktail of peptides, you can see an increase in CD69, CD25, and interferon. If on top of that, we treat the cells with nivolumab, we see a further enhancement of T cell activation as expected. This is the checkpoint blockade working essentially. If we then treat these cells with RIPR PD-1, using the same scFv as nivolumab, you can see a further enhancement in T cell activation. And we can see that, again, looking at all these three markers. We also wanted to control for CD45 dimerization. So what we did was we generated a diabody where the two arms bind to CD45 only. So there's no binding to PD-1. And essentially, it looks like dimerizing CD45 is not enough to see this effect. So in one hand, nivolumab is dimerizing PD-1, CD45 diabody dimerizes CD45, and it looks like by dimerizing both CD45 and PD-1, that's when we have the most robust type of increase in terms of T cell activation. So after this, we wanted to move to in vivo studies in mice. And for that, we had to develop a mouse RIPR. It's essentially the same molecule. It still binds to mouse CD45 and mouse PD-1, with only one difference now in that the binding to CD45 is mediated by an antibody instead of an scFv. So as you can see, we produce this molecule. Again, it looks monodispersed by SEC. We tested it in a very similar setup as before, but now using mouse T cells that were activated with an anti-TCR antibody. And then when we look at IL-2 or interferon, as you can see, if these cells are treated with just PBS, these are the levels of activation we observe. If we treat them with the mouse RIPR PD-1, you can see a potentiation of IL-2 and interferon gamma secretion. If we treat them with a blocking antibody for PD-1, we don't really see that. And if we block these cells, or if we treat them with an antibody for PD-L1, we also don't see this effect. So again, it seems that the mouse version of this RIPR PD-1 is also able to potentiate T cell activation. So the other thing that I would like to point out here is that, and that's mostly because of the architecture that we used, which now uses an antibody fused to an scFv. What that allowed us to do was to insert a 3C cleavage site. So this is a sequence of amino acids that is recognized by an enzyme, and then it's digested. So we generated this intact RIPR molecule or cut RIPR molecule, one where we still have the anti-CD45 and the anti-PD-1, but they're not fused anymore now. So we produced both molecules. And as you can see here by size exclusion chromatography, we have the intact RIPR in blue, the cut RIPR in red, and on the gel, on the protein gel, you can see intact RIPR around 37, running around 37 kilodaltons, and the cut RIPR separated or split in its two components, the anti-CD45 and the anti-PD-1. And now we could use this as a control to really stress test our model. where we could activate again T cells and incubate these T cells with intact RIPR or with cut RIPR. And here I'm just showing some FACS plots where we gated or we're gating on CD69 and CD25. And you can see this is the level of activation that we get when we incubate cells with 2 micrograms per mil of anti-CD3 or anti-TCR antibody. Around 16% of cells become fully activated. If we treat them with intact RIPR, this population jumps to approximately 35%. If we treat them with cut or digested RIPR, we go back to the same 16%. So essentially, it looks like treating the cells with anti-PD-1 and anti-CD45 separately is not enough to have this RIPR effect. They need to be together. And here in the bottom, I'm just showing the quantification for a few different concentrations of RIPR molecules. And as you can see, treatment with cut or digested RIPR is not enough to potentiate the response. So with this in mind, we then decided to move to the actual in vivo experiments where we decided to test a few different tumor models. So the first one is a pancreatic tumor model where we inoculated mice with this cell line, KPC. As you can see, in the absence of any other treatment, these tumors grow over time. They grow very fast, around 20, 30 days. They achieve quite a large tumor size. If we treat these mice with anti-PD-1, we can delay tumor progression. And as you can see, 2 or 3 mice out of the 5 do seem to show reduced or delayed tumor progression. When we treat mice with RIPR PD-1, 2 mice became tumor-free and 1 mouse showed slow tumor progression. And in 2 mice, we didn't really observe an effect. Now, this was a pancreatic tumor model. So then we moved to a different type of model, this colon adenocarcinoma model, where we inoculate mice with a different cell line, MC38 cells. And again, you can see in the absence of any other treatment, these tumors just grow over time. If we treat these mice with anti-PD-1, and this is out of 10 mice, we can see that 2 mice show a delayed tumor progression when they're treated with the antibody. When these mice are treated with the RIPR PD-1, again, you can see that 2 mice became tumor-free and 1 mouse showed a slight tumor growth delay compared to the other mice. So now we wanted to actually characterize a little bit better the immune response, particularly focusing on T cells, and try to compare if there's any differences between treatments with anti-PD-1 and treatments with RIPR PD-1, both in terms of T cell activation and T cell exhaustion. So for that, what we did was a shortened version of the MC38 tumor model, where instead of letting it run, let the tumor grow for 20, 30, or 40 days, we actually reduced this experiment to 12 days, mostly as an attempt to try to profile T cells at the peak of their anti-tumor response. So for this, what we did was we inoculated mice with MC38 tumor cells. We started treatment after day 5 when tumors were still rather small. We injected mice, or we treated mice with 3 doses of either anti-PD-1 or RIPR PD-1. And on day 12, we sacked the tumors and also the spleen and lymph nodes. And then we characterized T cells in terms of their activation and exhaustion markers. So first, the data that I'm showing here is for tumor weight. And this is just to show that even on this shortened time frame for this MC38 tumor model, we can already see that mice that were treated with RIPR PD-1, they have a smaller tumors compared to untreated mice or anti-PD-1 treated mice. And then when we look at markers of T cell exhaustion, such as CTLA4, TIM-3, or LAG3, present in T cells that were isolated from the tumors, so these are tumor infiltrating lymphocytes, what we observed, and this was surprising because we really weren't expecting to observe this, what we find is that in mice that were treated with RIPR PD-1, the percentage of T cells that are positive for CTLA4 or TIM-3 or LAG3 is reduced compared to mice that were treated with anti-PD-1. So it looks like overall, the presence of exhaustion markers is reduced on these cells. And here I'm showing data for CD8 T cells, but we observed a similar effect on CD4 T cells. On the other hand, when we look at markers of activation, such as CD62 and CD44, and in particular at this population of effector memory T cells, it looks like T cells or mice that were treated with RIPR PD-1 have a higher fraction of effector memory T cells. So this is a population of T cells that has CD62L low and CD44 high. The difference between untreated or treated with with anti-PD-1 is not really substantial, but it looks like treatment with RIPR PD-1 enhances this fraction of fully activated or effector memory T cells. There's also other markers or activation, or in this case, markers that suggest that these CD8s may be leaving sites such as the spleen and going to the tumor. Here you can see again that mice that were treated with RIPR PD-1 show an enhanced CXCR3 expression compared to mice that were treated with anti-PD-1 or that were untreated. So summing this part, it looks like treatment with RIPR PD-1 not only leads to a decrease in tumor growth, also leads to a decrease in the expression of classic exhaustion markers, and at the same time, an enhancement in T cell activation. So then we wanted to essentially step back and test how far or how much could we apply this system or this approach to different receptors. So the data that I'm showing before, that I was showing before was just on PD-1, and we wanted to see whether we could use a similar approach to target other receptors. So again, we went back to the HEC-293 reconstitution experiments, and what we did was to express LCK plus several different receptors, such as TIGIT, CTLA4, CD28, TIM3, common gamma, CD5, or B7H3. And as you can see, in all these receptors, in the absence of LCK, we observe no phosphorylation. When we co-transfect with LCK, we can detect an upregulation of phosphorylation for these receptors. And then on top of LCK and the receptor, we can also express CD45. And then when we do that, as you can see here, expression of CD45 reduces phosphorylation for most of these receptors. So what this is telling us is essentially confirming or suggesting that CD45 can act on many different receptors. And potentially, if we were to develop RIPR molecules for some of these receptors, perhaps we could be able to reduce their phosphorylation by recruiting CD45. So that's exactly what we did for a few of these receptors. So here I'm showing data for T cells that were treated with a RIPR molecule that targets mouse CTLA4. And as you can see here, again, this is the classic activation experiment of T cells plus anti-TCR, gating on CD69 and CD25. We can see here 13% of cells in these conditions become fully activated. When we treat them with RIPR CTLA4, we see an enhancement of the fraction of T cells that express these two surface markers for activation. This is true for CD4s. We also observed a similar effect for CD8s. We can also essentially move away from T cells and try to target receptors outside T cells, such as, for example, SIRP-alpha. Now, SIRP-alpha is present on macrophages and plays an important role that has also been described as the don't eat me signal, in that when it binds to CD47, essentially, it generates an inhibitory signal in terms of phagocytosis. So what we did here was to generate a RIPR SIRP-alpha molecule, which can bind to SIRP-alpha and to CD45. And we set up a very simple phagocytosis assay, which is a co-culture of human macrophages incubated with a target cell, in this case is a B cell, that it's treated with rituximab, an antibody that binds to the B cell, and that it promotes phagocytosis. So when we leave T cells or these macrophages untreated, we observe approximately 40% phagocytosis. If we treat these cells with AB21, which is an SCFV that binds to SIRP-alpha and blocks it from interacting with CD47, we can detect an enhancement of phagocytosis. And then RIPR SIRP-alpha further enhances phagocytosis. We can also look at the phosphorylation of the receptor. And again, using a similar experiment, what we did was to IP SERP-alpha from the surface of these macrophages and look at phosphorylation levels of SERP-alpha, either untreated or treated with the blocking antibody, which doesn't really seem to affect much the phosphorylation levels, or treated with RIPR SERP-alpha, which seems to lead to a reduction in phosphorylation levels. And then lastly, we also developed a RIPR molecule for CD28. Now, CD28 is different from PD-1 and CTLA-4 in that it potentiates T-cell activation. So here, we're shutting down, or we're trying to shut down signaling from a receptor that usually potentiates signaling. So we expected to see the opposite phenomena that we observed before. So again, we set up a T-cell activation assay. Now we're looking at CD25 and CD44. Around 76% of T cells become activated under these conditions. If we then treat them with a RIPR molecule that targets CD28, we see a reduction in T cell activation. And here you can see the quantification. With increasing dosing of RIPR CD28, we see a further reduction in the activation by looking at markers such as CD44 and CD25. So to summarize, essentially, it looks like some of these receptors, such as PD-1, can signal in the absence of ligand binding. This is also what has been called by tonic signaling. This tonic signaling can affect T-cell responses. And then molecules or bispecific molecules that target in CD45 and PD-1 or SERP-alpha, for example, they can reduce the phosphorylation levels of these receptors. They can potentiate T-cell and macrophage activity. And in vivo, in tumor models, they seem to be able to reduce tumor growth. So all the data that I've shown so far is essentially using off-the-shelf components. So the scFvs that we were able to find looking at the patent literature mostly. But it's possible that these molecules can be tuned, and it's also possible that the molecules that we developed may not be optimized in terms of potency. So for that, we can try to change or to check whether we can tune these molecules by changing the epitopes that they recognize, by changing potentially the distance between the two binding arms, by changing affinities, and we can explore signaling by other receptors by potentially changing the target, not only the target of the receptor, but also the phosphatases. Obviously, receptors such as, or phosphatases such as CD45 and CD148 are widely abundant in lymphocytes, but they're not present in many other cell types. But in the human genome, there's around 21 of these tyrosine receptor phosphatases. And potentially, of course, not all of them will have the same broad specificity, but we could potentially imagine using some of these phosphatases to target other receptors that are present outside the immune system. So essentially, to complement these two other approaches that have been used, such as the engineering of natural ligands and antibody blockade to modulate receptor signaling, what I've shown here is an example where essentially we used an enzyme-driven approach, in this case a phosphatase, to target signaling by these receptors. But again, we could imagine that maybe a similar thing could be achieved by using kinases or proteases or ubiquitin ligases. So phosphatase is just one of these enzymes that are present on the cell surface, but potentially we could use many other types of enzymes or other types of enzymes to affect signaling by other receptors. And with that, I would like to thank by thanking Chris, my supervisor at Stanford, Leon in his lab for his tremendous help, and everybody else as well in the lab for all the help. And then collaborators at Stanford, Julian Sage, Ron Vale at UCSF, Mike Dugan at MIT, Crystal Mackall also at Stanford, and Calvin Kuo as well. And thank you for having me here, Erdinch, and I would be happy to take any questions.
Thanks a lot, Ricardo. Fantastic tool. That's really, really nice. So I will start with a few of my own questions. So first, of course, you showed for a few proteins, right? So CD45, also oppositely CD28, and a few others. So how easy to make this kind of RIPR constructs?
They either express extremely well or they don't. That's usually the first problem because this bispecific diabody format sometimes produces proteins that are not very stable. And it's difficult to anticipate that, or it's difficult to predict what's going to be a stable bispecific diabody and what's not. It definitely looks like working with nanobodies is easier. They tend to be more stable. And it's much easier to get higher yields as well, which can become very important, particularly for in vivo experiments. But then there's also the question of how many times does this actually work? Now, we don't know exactly, or we have very little numbers for that, but so far we have these molecules for mouse and human PD-1, for mouse CTLA-4, CD28, and SERP. And in all of these cases, it looks like it worked. Now, how dependent is that on the binding epitope to CD45? We don't know, or to the phosphatase, we don't know. But I would imagine that not all epitopes will work, and that some places in CD45 around the phosphatase will be more easier, or will produce more potent results than others. And we don't know that. We don't know the answer to that yet because we haven't done a systematic approach, or we haven't used a systematic approach to scan different epitopes on CD45 and see whether there's any dependence between the epitope and the effect that we're observing. But this is something that we would like to do, of course.
Great, great. So related to this, the epitope that you're aiming in, let's say nanobody. So in the nanobody, first of all, to understand precisely, you have single binding domain. So you have basically two single binding domains in the RIPR construct.
That's right.
So can you engineer the single binding domain that binds to the phosphatase so that it doesn't bind only to CD45, but also other phosphatases like CD47 or CD148?
Potentially, yes. Yes, potentially. Yeah, potentially.
Do you think it would be useful so that you get rid of all the phosphatases?
Potentially. We could think of ways to generate some of these molecules, I agree. Yes. We should be able, nowadays, so to generate these nanobodies in the past, you'd have to immunize a llama, so you would need someone with an access to a llama. But nowadays, there's yeast libraries and phage libraries available that make the process much easier to identify some of these nanobodies, of course.
Well, it's only llama, don't worry, Ricardo, we'll send you some. Okay, first I'll ask some questions from the audience and then I'll come back. I still have many questions for you. So the first question is from Ardakan Üner, and he's asking if you can use this for autoimmunity. You showed instead of increasing the T cell activity by having RIPR against CD28, you can actually decrease the activity as well. So what do you think that's potential for autoimmune diseases?
It's possible, of course, and that was the aim with the CD28, was to essentially show that we could also try to develop ways to decrease T cell activation. Now, one thing that we observed in making these molecules is that, of course, we anticipate these molecules work in cis, but it's important to think that they can also bind in trans. And we know that, for example, antibodies for TCR will activate the T cell receptor, but an antibody for PD-1 usually does not activate PD-1. And I don't think we know why. And the same thing is true for CTLA-4. So an antibody for CTLA-4 many times does not activate CTLA-4. So this to say that for some receptors, binding in trans activates them. And if that's the case, that could mean that in a RIPR molecule, because it's possible that it can bind in cis, but also in trans, we may have a mixed result. We may have inhibition by binding in cis, but also activation by binding in trans. So we tried, for example, some RIPR molecules for TCR, but they didn't really work. And we think it's because some of the molecules could be binding in trans and activating T cells in that way. So in theory, yes, but in practice, we need to understand a little bit more about the effect of the trans signaling and choose the targets carefully.
Thanks, Ricardo. So the next question is by Devrimus. Can PD-1 receptors make dimer or oligomer? And have you checked T cell stimulation by dendritic cells?
We have not. We have not. So to the second part of the question, in the PBMC assay, we were relying on peptides to activate the T cells. And because it's a complex mixture, it's possible that there will be DCs there, but we don't know for sure. But the activation was driven by the peptides. Now, and we haven't really tested just a DC co-culture with the T cells, essentially. Yes. Oh, and the first part, what was the first part again of the question?
The first part is if PD-1 receptors make dimer or oligomer.
It doesn't seem to be the case. It seems that PD-1 is mostly a monomer.
Okay, thanks. Next question is by Christina. My question is, may the RIPR binding change in the PD-1 resistance? Additionally, does RIPR binding induce intracellular events such as apoptosis or phosphorylation of other receptors?
That's something that we would like to look at, of course. Now, the next steps will be to look at that, to see what are the effects in the rest of the signaling pathways that T cells have. We don't know yet.
Okay, thank you. Next question is by Eje. Could RIPR PD-1 in your study affect cytokines such as interleukin? Could it be possible effect on the interleukins alter cell function?
Possible. So we observe this potentiation of cytokine secretion, such as IL-2 and interferon. And it's possible that they will, of course, induce changes in cell proliferation and differentiation away from the actual T cells. And that's probably potentially true for interferon that we know has an effect on many different cell types. So how this changes and how this affects the environment, we don't know yet, but it's possible. Sorry, Ardinch, can you repeat the question? I think. You appear frozen on my screen.
Can you hear me now?
Now I can hear it, yes.
Okay. So the question is by Mezida Said, and she's asking, does the ligand-independent tonic signaling by the inhibitory receptors always require activation through the TCR and LCK, or does similar effect occur with cytokine or non-specific stimulation, such as PMA?
Right. So that's a very good question. So we also, I didn't show here, but we also tested activation or treatment with RIPR in the absence of T cell receptor stimuli. And there, RIPR didn't really potentiate signaling. So it looks like we need a T cell receptor signaling to observe this potentiation. Particularly if we're looking at cytokine secretion and upregulation of markers such as CD69 and CD44. But that said, it's possible, of course, that if we reduce tonic signaling of PD-1 and if we reduce the amount of SHIP1 and SHIP2 that is present at the membrane, by doing so, this may change signaling by other receptors, including TCR, but it doesn't seem to be enough to drive a robust T cell activation response.
Mm-hmm. Thank you, Ricardo. Can you still hear me?
Yes.
Okay. So one question. So now what you showed is always in the extracellular space. How difficult is to make it intracellular?
Well, it's the membrane. It's the membrane. There are reports. There's been some studies showing protacs. I think they're called protacs. So small molecules that essentially do a similar thing, where they can join or they can link together an enzyme to a target, and these, of course, can also change signaling by those receptors. But these have mostly been small molecules that can cross the membrane. Now, with things like adenovirus, we could think of ways of expressing intracellularly some of these bispecific diabodies, but we haven't done that yet.
Okay, great. So next question is by Hussein Amredindar. What is the duration of RIPR inhibitory effect on T cells? And what is the type of inhibition, competitive or irreversible?
So we looked for up to 5 days, and we still saw an effect. So this was treating the cells on day 1, and then measuring activation levels up to day 5. And we still saw an effect on day 5. What we haven't done is the second part. So we haven't stopped RIPR treatment or removed RIPR from the cells, and then checked whether these cells could be restimulated again, and what would happen in terms of restimulation. We haven't done that second part, but that's a very good question.
Thanks, Ricardo. Next question is by Betul Köklü, and she's asking if this RIPR treatment could cause actually autoimmunity and high reactivity.
Right. Yeah. That's of course that would be an issue, and that's possible, particularly because we already know that anti-PD-1 and anti-CTLA-4 treatment have some toxicity, and they can induce autoimmune responses. And one would expect that the same thing could happen for RIPR. Now, one possibility with RIPR is that we could potentially target different CD45 isoforms, and that's just a theoretical possibility. We haven't done that. But it could be possible to target different CD45 isoforms. And the advantage of doing that is that we could direct the RIPR response to either naive T cells or activated T cells, because these different T cells express different isoforms to different levels. So it could be possible to modulate some of the RIPR activity, or at least to direct it to particular T cell types. But that has to be tested, of course.
Thank you, Ricardo. So the next question is by Rita Santosh. Great work, Ricardo. Congrats. This will be a super elegant way to target orphan receptors, which function relies on only tonic signaling. Do you have any data on this already?
Not yet. But that's we think that could be the case, and you know, there's many receptors out there that don't seem to have a ligand, or if they do, it has eluded you know, decades of experiments to try to identify them. And it's also many times it's also unclear how some of these receptors signal, and maybe they signal by tonic signaling, and that's it. And of course, antibody blockade probably doesn't really do much for those receptors in terms of signaling. So the RIPR approach could be something to explore.
Thanks, Ricardo. Next question is by Ashraf Mamadov, and he's asking, you showed that RIPR PD-1 causes CD8 memory increasing. Is this mechanism of this alteration is certain or is known? We
don't know. We now would like to characterize these T cells a little bit better, sequence do some exome sequencing, do some metabolism analyses, and try to characterize these better. We don't know yet.
And next question is by Irem Kulu, and she's asking if RIPR PD-1 can be tried on other solid tumors other than SCLC. So is there a reason why you tried SCLC? And on top of this, I am adding one more section. Did you also try, let's say, melanoma, where actually the PD-1 blockade receptors really work well?
Yes, we haven't tried melanoma, and it's something that we should and we plan to do. When we tried SCLC, was really a chance event in that Julian Sage, who helped us with those experiments, is at Stanford and was kind enough to allow us to test our molecules in his tumor models. But the lung tumor model also had an interesting twist to the story in that it mimics relatively well what happens in humans in that the tumors respond to chemotherapy. And then also respond to anti-PD-1 treatment. So we wanted to see whether co-treatments with RIPR and chemotherapy would improve or not the anti-PD-1 or the RIPR effect. And it looks like that was the case. In that particular model, RIPR PD-1 alone wasn't really inducing much of a response beyond what anti-PD-1 does. But in combination with chemotherapy, it seemed to elicit a more robust or a more potent response.
Okay, Ricardo, thank you. So now, last question by the audience is from Jansu Özenen. Is it possible for mesenchymal stem cells to self-destruct endogenously before RIPR PD-1 reaches the tumor cells? Do they express these proteins?
I don't know. That's a good question. I don't know. I'll check. I need to check.
Okay, so I have last question for you. So now you showed membrane. I asked intercellular, especially nanobodies would be probably possible to do intercellular. But let's say, is it possible to click two soluble proteins in the extracellular space?
You mean to have two separate nanobodies and then click them somehow?
Click two, let's say your proteins are not membrane bound, but these proteins are secreted proteins. You want to click two of them together in a 3D environment, not in
right. Yes, I think it should be possible. I think so.
I think that would be also useful for, I don't know, like applications in development or other things.
Sure. I think so. The limiting factor is always finding good binders, good stable binders, either nanobodies or SCFEs. Once we have those, then I think we could do these sorts of things. Yes.
Wonderful. Thanks so much, Ricardo. That was a great pleasure to have you. Great talk, fantastic technology. I'm really looking forward to doing some work together.
Thank you. Same here. Absolutely. Thank you for having me, and thank you everybody for listening.
Thank you very much. So see you soon.
Yes, see you.